Hi, and welcome. This is going to be on heart formation, and I'm Gavin Fitzgerald. Alrighty. So, to get started, we have a couple different things. Obviously, the heart, as an adult, we have like an image here, and so that's going to have your 4 chambers. For this presentation, I focused mostly on the human heart. The research is mostly on like mouse though, but it's both mammalian. Even though everything has, well, most everything has a heart, so we have different types, obviously. But the important like background information about the heart development. So the heart is very important for oxygen, blood, and like nutrients to circle the body, get to all the cells and the tissues. And then also, there's a couple fun facts. I love fun facts. So the adult heart, for at least for humans, pumps about 2,000 gallons a day, which is a lot of stuff, a lot of blood. And then another thing, the heart beats about 115,000 times each day as well. So just massive numbers from this rather small organ. To think the average heart is the size of a fist in an adult. So it's a pretty small organ, but it has a lot of importance, and it does a lot of things. So the image here on the screen, hopefully you guys can see it, is the human heart. It has the nice 4 chambers, so it's got kind of reading from left to right and from top to bottom kind of thing. We have the right atrium, and then we have the right ventricle, and then the left atrium, left ventricle. So those are going to be the pumps. And it's pretty interesting. And there's a couple more facts on here. So if you want to just read those, you can for sure. It's complex looking, but it's not too hard, I guess. So first, second off, I guess, let's talk about some other species hearts. And so, like I said, most of this presentation is going to be focused on human heart. So I thought I would obviously throw in the other types of hearts that we have. And there's a couple, at least, right? So we have the 2, 3, and 4 chambered hearts. So, and they're roughly the same they're roughly the same because it's just a system that's being pumped through. The only difference is it's more complex because they're more complex creatures. For example, we have the 2-chambered heart for the lizard, and also the fish has the 2-chambered heart as well, and then 3-chambered, and so on and so forth. Actually, I believe the lizard is a 3-chambered heart. Sorry about that. But all the way up until the mammals, which is going to be the mouse, the human, things like that, with that 4-chambered heart, which is what the research later on that I'm going to be showing is also focusing on. All right, and then next up. So, I'm not really sure how this audio recording thing is working, so I hope it is working now. We had some issues earlier, so these slides might be slightly text heavy, but that's just in case nothing works. And so to start out, we have the pair of chords. These are going to be the cardiogenic chords. And from that, we are actually going to get a hollowed out, pretty much 2 tubes. So a pair of tubes. And it's amazing that these things almost immediately start. So we have, what is it here? We have day 21 and 22, it starts pumping, which is remarkable. That's like 3 weeks in, roughly, after fertilization. So to think this thing is already like pumping and beating and everything so early on. Now, it does get a little intense. We have the mesoderm, and then that's going to be inside of that trilaminal embryonic disc there. And the next slide I have more images that I'll be able to go in depth and everything, and I might flip back and forth here in a second as well, just so you have something to kind of visualize depending on what style of learning you are. So, and then what's going to happen is in that pericardial cavity is where these paired tubes are going to be. And then when that disc nicely folds up, it's going to get carried to that correct location in the chest cavity. So not everything yet is formed, but it is kind of folding up and bending every now and then it's slowly moving to where it's going to be in the finalized body, because obviously an embryo does not look like an adult human yet. More like it looks like a little alien kind of creature. And so after that, what's kind of happening is those two pairs of tubes are fusing, and then we have all the fancy words here that saying they're fusing, and then they're going to differentiate into the like 4-5 kind of compartments, and those are going to be the truncus areatosis, the bulbus cordis, and the primitive ventricle, and the primitive atrium, and then also that sinus venosus. Venosus, yeah. This happens close to around day 22, is that these things are kind of being formed in there. And then what happens is very weird. It's kind of crazy, but these tubes start forming like an S shape, and it's kind of forming an S shape and then bending and curving as well. And that is inside of the pericardium. And that's going to be right after all of those different compartments have formed. So between 23 and 28 days that's going to be going on. After this, around near 28 days, that internal septa is going to start forming. And that's going to separate the heart into those two main sections, the atria and the ventricles. So those two main kind of compartments. Now, a little later on, 5 to 8, 5 to 9 weeks in, so a good ways down compared to like 20 days. So now we're about 5 to 8 weeks, 5 to 9 weeks down. The aortoventricular valves are going to start forming, and that's what's going to help with that blood flowing in and kind of actually getting that blood circulating really well, and so that there's no backflow and issues like that, because the human body is very smart in that way, you know. And then so once those valves form, that's going to be the same time as well as those semilunar valves are also forming and formed. So both of those valves are helping with that blood flow, and we'll see those in pictures here in a second. So so definitely, I know it's a text-heavy slide, lots of stuff kind of crammed in, but it's very simple, as I can just write it out in a paragraph, but it's actually very complex. So we'll see now on this slide here, we actually have a couple different illustrations of it, and it gets a little crazy, but don't worry, it's interesting. So on this first one here, and I'm doing left to right here since I'm not actually there pointing at it, we have those two cords there, and that's going to become the two tubes and everything. And then as we go down, you can see them fusing there, and then once they fuse, it kind of shows the different areas of like so that first one's very nicely color coded, so you can tell what it's going to be in the end. So we have that nice truncus, what is that, areatosis, I hope I'm pronouncing these words right, and then the bulbous cords, and the primitive ventricle, and the primitive atrium. Now those haven't actually divided into what we know as like the ventricle and the atrium, so that's why they're the primitive ones, because they really aren't, they're not what we think of in like the fully formed heart that we normally see when we look up, hearts. And then it kind of gets to this nice S-curve and weird little twist. So it's very complex here. These next like 3 images there, a lot happens. So we have that nice S form, and you can see that by that red part getting pushed in. and then that S part kind of backs out a little, but then it kind of swings around and hooks up near the top, so that those what do we have here? The aortic arch arteries are right above the right atrium and the left atrium, so that primitive atrium kind of wraps around, and then it divides into those right and left like categories, those right and left compartments. There we go. And then the next, and then farther down you can see that things are partitioning here, and then you can see the septum premium is forming, and then you'll have those nice chambers in there at 28 days, and then at 8 weeks. By then you'll have nice, more formed chambers, because remember, this is a 4-chambered heart. All right, and so now this next one here with the red and green, it's like the little Christmas tree here. It's relatively the same thing that's going over here. It's just a different look at it. So you can see more of that S shape, and then also you can see more of how it kind of curves and then bends up. And this is really hard to explain because I like moving my hands. So without that, it's a lot harder to explain with just looking at these pictures. But it is just an S form, and then it kind of does that bendy thing up. And so that's why I put this other image here, so that you can kind of see it looking at it as if you're looking at the actual S and then the bend. Because this other image just kind of has it to the back, so you can't really tell. And then this final image here, this is going over how it's going from the neural disc, like the embryonic disc and everything, and the neural fold, and then that's going down to show how you're getting that the heart development that way. So I know on the last slide here, if I switch back, I think that'll switch back for you guys as well. Here you can see, what is it here? Yeah, in the trilaminal embryonic disc, and then that's going to be in, hold on, oh man, it's in that last paragraph, I believe, there. And so that's going to be that folding and everything. And, it's in the middle paragraph, when it folds and gets carried to the correct position. So what's happening from the top is it's folding in and then kind of jutting out, and it's a nice slice. So it's a little hard to comprehend because it's not how you normally look at the heart. And so you can see those heart-forming regions. Once they come down, they're shooting out a little bit and then bending out. And then the forming heart, it's labeled on there, so it's that nice little red pocket there. And what that's going to do is it's just growing outwards, and then it's creating those little circles there on the bottom, right there. And so that's going to be the heart, because there's the gut and then the heart. Well, the foregut, so not really the gut. Very weird. But it looks a little weird, but it's just another way, depending on how you visualize things and everything. All right, so now this is just another brief summary of it. So we have the heart forms from those 2 tubes, and then it creates that 4 chambers. And so then again, since I didn't know, or I'm not sure if this audio is going to work, I have on there a little label there, so it kind of shows what's going on. So let's hop in. And I like these 2 images here. So for those of you guys that are going on, maybe you're going to become heart specialists or something like that, we do have the proper stages and labelings here. And so both of these have the same labelings. And then we also have a comparison on the what is it here? The more right one. Again, without being able to point, it's not very fun. We have 2 different versions here. We have the human, and then we have the chick. And it's kind of comparing the developmental stages. And I find that nice because we've covered the chick, obviously, so it relates back. But then also, it shows that there are differences in development, even though there are a lot of similarities as well. because you can see that we, in the beginning, still have that same bend there, and as well as the chick. Mind you, the chick is a lot faster in development because it doesn't take them a long time to develop. It takes them, 20, 21, 22 days, versus us, it's like, okay, we have a couple months. So the times are obviously different, but the development itself is not that different. They also have a different number of chambers. I believe it's 3 chambers. But 3 or 4, maybe. But then, yeah, so we have similar development stages. It is a little weird that they put it in more of like image looking, and then we have ours in like almost like a labeled version. But I thought it was cool to just see that comparison there. And then on the other side, I really like, on the left side, sorry, I say other side, but yeah, on the left side, we have a really cool one that I like. Because not only does it label everything here, like the outflow track and the inflow track, and then we have there the formation and the curving. So what's happening is, once it curves like that, you can start to see that S forming, and even when that S is forming, like I said before, those atrium and ventricle and everything are actually forming there, so we can see like, what is it? Like in the embryo, when we look at it and we can tell where things are going to end up, usually because of experiments previous, it's the same way here. We can actually tell where things are going, which is really cool to see that everything is already slowly knowing where it needs to go and what it needs to do and everything like that. So, that being said, as we go to the second line here, we can actually see that those chambers are already forming even more pretty soon. So as soon as it starts looping around, those 4 chambers are there. Like they're the right, the left atrium and ventricles are pretty much formed there. They're not fully formed, but they're there. So by the time that it's fully bent around, you can actually see all 4 of those chambers. As well as the inflow and outflow. And I like this diagram simply because it kind of simplifies it, and then also shows you can actually see like the bubbles and stuff that are actually going to become the chambers and everything like that. Alright, so let's go on here. And so now we're going to be talking about the research. And again, some of these first slides are a little text heavy. I wanted to be able to encompass everything that I could. It's a very interesting paper, and the first thing that actually drew me to this paper was the images, and they have these videos, and I'm not sure how, I don't think I can show them to you on here. I might try, but it might not work. But they have some very cool videos that I really recommend farther down when we get into the data, that you click on and you go and look. Even if this does not interest you at all and you really don't like heart formation, the images are very cool, how it shows the ups and downs, and then also just the colors. And I don't know, some of you might not be like people that like colors, but I love colors, especially in diagrams and everything. So these are really cool to see. So yeah, let's dive right in. We have cardiomyocyte orientation modulated by the nub family proteins and coherent access, and it's essential for that ventricular wall morphogenesis. Now, don't worry, when I first read this, I was like, what? And that's perfectly normal, because, again, we're just learning this, Unless you have taken these developmental classes earlier on, it can definitely be interesting and hard to understand. So, what it's talking about is it's going to see if those nub family proteins, if they really are, first off, essential for that wall morphogenesis, so that wall kind of creation, in another sense. And so, yeah, it's very, very fascinating. They did a lot of knockouts and knock-ins, and they tried to correct what they messed up and fix it, and it's very interesting. So, and I'm just going to skim this paragraph. If you want to, that's why I put it in there, so that you can read, you can go through it. I also put a link in, I think, on the next slide for if you really just are fascinated by this article. It is a cool read. It's a little difficult, but it is very cool. And they have a lot of great images and all kinds of things like that. So that, what do we have here? The central heart formation, so that's going to be that trabecula in the myocardium is it's very central to that heart formation. So what's going to happen is that increase in mass, that muscle mass, before the formation of that coronal circulation, so before the blood and everything, and before it starts circulating, it needs to have that big muscle mass grown. And that the cardiomyocytes are in that word, spatiotemporal orientation during the trabecular formation. Of course, we couldn't have named things simpler. And then also those nub and nub-like double knockout display abnormal cellular orientations. So that's pretty cool that they were actually able to see something happening and everything, because that always makes science more fun, right? And then that the knockouts displayed reduced membrane localized N-cadherin due to that attenuated N-cadherin recycling to the membrane. Alright, and so when things are overexpressed in the knockouts via a mosaic model, the defects of the cell orientation were there, and they were also they could partially rescue them. Alrighty, so let's go to the next slide. And again, this is just part of their abstract here, but and it's just here in case you really want to read it. It does go over pretty well, but just like this one over here and this one, they're both relatively the same. That one is more of like, why is this one so great? And this one is more of like, what did we do? But either way, and then there also is that link in there. It should be hyperlinked if you want to follow it and go into more depth and detail and fully read this and learn everything. So they're really looking at how nub regulates and regulates cell orientation, behaviors, and different things, and it'll be determined using those N-cadherin levels. Yeah, alright, so here we have all the figures, and like I was saying, they're really cool figures. They have lots of stuff, and lots of colors as well, so it makes you kind of like, ooh, what's that? More curious, you know? So yeah, and again, these will come back later when they're using them to prove their different things. Alrighty, so here we have some more figures. One important one, let me see here, I believe it's number 3 here that's important. But they all are used to prove their results and everything, alright? So, there's that. And I like number 6 just because colors. But it also shows a lot of stuff as well. So, before we get to the results here, since this is not a class lecture, like I was saying before, those videos and stuff that I wanted to show, all the videos are hyperlinked that they have in there for their supporting their findings and everything. And I do recommend watching them, either because they're cool. Some of them might not make much sense to you if you don't. You might want to read the article as well, but they're pretty cool to just watch as well. But yeah. And then let's see here, we have the corresponding videos also on all the slides, as well as the figures and data and everything. And so it's broken down right after this next slide. That's the results. Alrighty? So, cool. So these are the results here. We have different things that they found, and these are like the main ones as well. I believe there is another one that did pop in later down the slide, but it wasn't one of these main ones, I don't believe. So we do have that. Alrighty? So here we have, and so they all start with NFPs, obviously, and that's going to be that nub proteins, if we remember, very back here. And that's going to be those nub family proteins. Alrighty, in case you didn't remember that. Which happens. It's cool. So those NFPs are required for cellular orientation and organization during the trabecular and ventricle wall morphogenesis, which is what they kind of set out to prove. They wanted to see if it was required and if it helped and everything for that ventricle wall morphogenesis. And they were able to see that using different expressions and things like that. Alrighty, and then the next one here is going to be that the NFPs regulate OCD and directional migration. Sorry about that. Directional migration. So that's pretty cool that they were able to see that these nub protein, this family of proteins, was able to help determine like where things are moving to and where they're going. The next one is good. NFPs regulate transmural growth and cardiomyocyte proliferation autonomously, which is very interesting. So we'll get down there to where they have the figures and everything. I wish this was on a bigger screen so I could like get those figures kind of blown up. So I hope they can see them on your guys' screens because we don't have like big projectors anymore. And so NFPs also regulate trabecular morphogenesis through N-cadherin. So this is showing that N-cadherin and then that family of proteins and everything and how they all work together using that N-cadherin levels. Okay, alrighty. So NFPs also regulate the transition of N-cadherin from early endosome to recycling endosome and its trafficking to the membrane. So this is very interesting to see that we can see that transition of N-cadherin is also being kind of regulated by those NFPs as well, which is pretty cool. And then lastly, that N-cadherin expression rescues the defects of cellular orientation and trabeculation. I'm really bad at that word. In MDKO hearts. So that's going to be those messed up hearts. Alright? Okay, so let's get in here. Alright, so this first one that I really liked, and I probably won't go over all of them, just for the sake of time, and the fact that you guys can easily go in and look at all of the results. I have laid them all out in here for you, so if you do want to dive in more depth, I have all the figures and the videos and the link to the paper as well. Alright, so this here is going to show you that it is required. I wish I could zoom. I wonder, okay, if I zoom in, I don't know if this is going to show up on your guys' screen or not, but I zoomed in. So let's see if it works. Again, I've never done this. So most of the cardiomyocytes in that inner layer, in that compact zone, and you can see those because you can see the heart forming. That's going to be those little lines on the screen there, and there's that little nice box there as well. And then they zoomed in on the other side of there as well. So let's see. Let's see here. The perpendicular to the heart wall. Where was I? Sorry, I lost track here of what I was saying. Oh my goodness. Now, wait, I think the audio, alright, I'm back. Hopefully it works now. The parallel orientation to the heart wall. So, and as you can read here on the figure, it's showing kind of what they're doing in the figure, obviously. And so we have here the double-headed arrows indicate the orientation of the cell, and asterisks indicate no orientation of the cell. Alrighty. And I wish I could point. I can't really point, so it's hard to like go over this. Now, we do have, you can see the different labelings here. Where are the colors here? They should be somewhere. Yeah, here. So each line, that's going to be for the circle. Where is it here? I just lost it again. But yeah, it's all showed. So if I, whoa. Sorry, it moved on me again. And so it's showing you that in this first one, here if I zoom in again, I think I zoomed in. Alright, So on this first one here on the left, we have the normal right here. So we're having the heart, and you can see that there is orientation because there is the lines, and there is the like red line and the green and the blue, and everything is kind of where it looks organized. Even if you have no idea what you're looking at, it looks organized, right? Now see, when they did it on the other side and they messed it up, you can see that there is really, there might be organization, but you can't really tell. It's just a big blob of things, honestly. And you can see the green, and there's a lot more green. And then the blue is kind of spilling out over here on the sides at the bottom. And it's more blob looking and kind of like, So, and that tells you that it is, that NFPs are required for that orientation and that organization. And that's simply shown here by the nice kind of organized looking versions where there's lines and everything's kind of fit together versus the other side where it is a lot of blobs and everything. Alrighty, and so this is, I really like this one. And then let's see here. We have the migration here. I'm not sure, if I zoom in, I don't know if that'll help you guys. I don't know if you guys can see that. And again, I wish this was on a bigger screen, then I could actually zoom in and point to things as well. So, and I'm not really going to go in depth on this one, just because, I mean, it is important and it is helpful towards the whole organization and migration of things as well. The growth and those mitocytes proliferation autonomously. And so you can see that using the different colors here, and then they also have quantified it in the graph there. Now a couple of these, they have a couple really cool videos that are like green, I think, and green and blue and everything, and it shows like the transfer and like, it's very cool on these. So these are some of the videos that I was talking about earlier that you're going to want to see. Now here, it's regulate this morphogenesis through like N-cadherin. And so, whoa, I try to move something and everything else moves as well. So here we have this N-cadherin, and you can see on the top that they see it there, which is a good thing since that's what they're doing. And then also if they, see they have the normal, they have the hetero, and then they have the hetero over there. And so you can see the different levels. Whoa, again, it's just moving on its own. I'm not really sure. And in the control, everything looks normal, which is always a plus. And then as we go down, you can see an increase. Let's see. On this one, you can see it's roughly the same, actually, on this next one here. But then when you get down farther, and you can see the N-cadherin in red, and then you can see less of it in the second bottom here, and less of it. And then over here, you see these big dots of blue. Let's see here. What? I hope it worked. So here we can see that where the nub is localized, and how that's localized in those endotic organelles. And I believe this is the one that was not earlier on, but I did like it, and it looked pretty cool as well. And so and this one, they haven't really proved this set, because it's a might be required for this. They don't really know, but they're getting it out there. If they do know, and it turns out that it is, then they can be like, we're the first ones that did this. But they can see that it is localized, and you can, whoa, okay. And you can see that in these images here. You can see that it's localized in those certain areas there. Alrighty? Yeah, that is very cool to see that they were able to get those images and everything there. Alrighty? And of course this was an antibody thing. So they used antibodies and then they lit it up pretty much. So they could see those colors. Alright. And so they can see here that it's fully recycling, and you can look through these pictures here. It's cool to see. They didn't have any videos or anything, but they did have the images, and you can see it lighting and darkening, and then going away as well when it gets rid of it. And that's going to be that N-cadherin. So, yeah, there's that. And then they have this one, the overexpression rescues it. You can see it's here by the end, kind of. But it does rescue it. So that's pretty cool that they were able to do that. It doesn't always work in things. So yeah. And then let's bring it back to that main thing. So that main starting question was, does that N-cadherin level, those nub family proteins with N-cadherin protein, the level of localization and everything, and it's cool to see that it does. It helps with that ventricle wall morphogenesis, and then it helps with that organization as well. And without that wall morphogenesis, there many defects arise with different like heart defects and everything because you don't have that nice separation between them. So you do need that. And another thing that I was seeing is that this paper is really cool because it's starting to figure out why things are working like this. So it's kind of digging in and it's seeing like what helps organize these things, what helps show these, what helps make these things happen. So like those nub family proteins help with the localization and everything. Because a lot of that isn't known that I was finding. A lot of that is still kind of up there. So they're still trying to figure out what proteins help and what levels of what really help with the development of the heart. Because we know what happens. Like we know the little S thing and it bends and the fold and everything. But we don't really know why. Like what's going on? And that's what we're trying to figure out. And this paper did it. It's really cool to see that it was able to do these things. All right. That brings it back here, and so that we're kind of getting both sides as well on this part. So earlier we have like what we do know, so it's like folding and bending and turning and everything and kind of forming, and now we're kind of getting that idea of what's going on behind the scenes. So what things are getting expressed to get these different formations and everything, and that's what I liked about this paper along with the heart formation. So hopefully you guys all liked it as well. These are going to be the links to the sources as well. So there's a couple different things. If you're really interested in it, there's some links